is the uh, the current received on the skin of a person when the heart undergoes the systole and diastole. Or you can also define it as the amount of the graph between um, millivolts and time with each systole and diastole of heart, or you can say with each cardiac cycle. So what happens that you may have seen this in the movies, that when we try to get the ECG, we place different types of electrodes on the skin. Have you seen it in different kinds of movies? It is very it is mentioned, it is like
Yeah,
discussed. Or
yeah.
for your interest, if you want to see a very good season, there is a season related to the medical field called as The Good Doctor. So you should watch it if you have free time. That is a very great season for the students to that are going to become doctors. That is a very great season, very great season. So in that season, it is also, you can say, a bit, you can get the a bit guidance about the ECG, electrocardiogram. So what happens that we place the electrodes and we get the following, you can say, readings there. This is the millivolts, and over here we have time. On y-axis we have the millivolts, and when there are millivolts, suppose that um, basically the electrical current starts at, you can say, the heartbeat starts at 0. Then there is one peak you will receive, then there is another peak after some time, and then there is last peak, up after which the next beat would start. So this is the total electricity against time in one beat. We divide each peak and each graph with, or you can say, we name or label each graph peak with different alphabets. This peak is called as P, then Q, R, and S, and the last curve is T. Now, what is the meaning of all these labels? Remember, P is the current during the atrial systole. The QRS is the current you are receiving during the ventricular systole. And the T is the current you are getting during the ventricular diastole. So that basically is the idea, that you'll be receiving this current when your atrial systole would happen, and QRS, you'll receive the current with the amount of QRS when the current is in the ventricular systole, and then the T is the ventricular diastole. So these different currents you'll be getting. So if you plot it on the cardiac cycle, the atrial systole, it is P, then this is QRS, and then this one is T. So this is T. So these are basically the different readings you'll be getting on the ECG. So remember that the ECG, what the ECG would tell you, first of all, total beats per minute. Total beats per minute, which is basically called as BPM. BPM. This is called as BPM, beats per minute. And how would you find it? Suppose that this is 0 second and one beat is completing around zero point 0.5 seconds. One beat is completing around, I'm sorry, not zero point 0.5 seconds. Okay, so yeah, let's keep it zero point 0.5 seconds. Zero point 0.5, then the next beat is completing around zero point 0.1, then again and again. So if you are given with the beats per minute, or if you are given with the beats per second, how would you calculate the beats per minute? So you would say that one beat equals zero point 0.5 seconds. So how many beats would be there in 60 seconds? In 1 minute, obviously, I guess you can easily um, you can like calculate it. How should I do it?
Multiply by 60.
Should we be multiplying it with the 60 or divide by 60? Sorry, 60 divided by zero point 0.5.
Yeah, 60 divided by 1. Oh, it's one beat. Okay, 0.5. Yeah, so
Yeah.
120.
So 60 divided by zero point 0.5. So that would be yes, 120. 120 beats per minute it is. So this is how you'll do it. First, you'll get the time for one beat. That is, maybe it's around zero point 0.6 seconds, maybe, or zero point 0.8 seconds. No problem in that. So if one beat is around zero point 0.8 seconds, then you can easily find that how many of beats there would be in 60 seconds. So 60 divided by zero point 0.8, you'll be getting your answer. And this is a question from CIEs. This is not just a random calculation. This is a question from CIEs. They ask us sometime for 1 mark or 2 marks that what would be the beat, and sometime they would be asking us in MCQs in P2. So that is basically an important thing. So always remember the ECG or heart rate can change. Heart rate can change. And what can change the heart rate? Like, obviously, can you name it?
You
Like
mean what
exercise?
changes the heart rate?
Yeah, heart rate or ECG. Like, obviously, ECG would also change.
Um, so obviously exercise.
Hmm.
Um, I think like, coffee.
Exactly. The caffeine is there.
I think I've heard tobacco also does it.
Tobacco also does it. One of the main thing. Let me give you this because this would basically help you. One thing is fear,
So
and
emotions.
another thing is amount. Yeah, amount of carbon dioxide in blood. It is one of the most important driving factor. When the carbon dioxide is higher, your heartbeat would rise. When the carbon dioxide is in low amounts, your heartbeat would fall. And that is basically controlled by the CO2 center in brain and also CO2 receptors in aorta. They are receptors in aorta that would be controlling how much CO2 should be there in the blood. If it is like other than if it is higher than the required amount, your heart rate would rise to get rid of the carbon dioxide, and this basically makes us panicked. um, do you swim?
Yeah, that also raises.
Like, do you go underwater?
Yeah, swimming.
Sometimes one of the most difficult tasks is to stay underwater while swimming. Why? Because your brain constantly is telling you that you are not releasing carbon dioxide. You
Yeah.
may would die. You have
Yeah.
to breathe. No, that is only panic. That is only panic. Your body always has sufficient oxygen, but since you are not very much, you can say, accustomed, you are not very much accustomed of holding your breath for the long time, so your brain starts panicking that you should be immediately releasing the carbon dioxide. One of the solution of this is to release the air, but that is obviously that has to be done with special training. But if you closely look at those divers, what do they do when they go underwater? You will see the bubbles coming out of their mouth.
Yeah.
Why? Because they release the air, and that would calm the brain a bit, that the carbon dioxide has been released. So that is how you should be controlling the panic of your brain. Anyways, other things can also change. They can come along with the. Now the next thing is the coronary heart disease. All right, can you tell me what is coronary artery?
Coronary heart disease.
No, no, coronary
Um,
artery. What is coronary artery?
it's the artery that just supplies blood to the heart.
Hmm. Like, why the heart would need the blood, by the way? Hmm?
I'm not sure.
If there is a person, there's a match going on, and there's a person who is running here and there to give the water bottles to the players. Does that mean that the person who is running here and there carrying the water bottles himself does not require water?
No, he would require water, but like not at that time.
Ex- exactly. The thing is, the heart does provide the blood to the other parts, but the heart also requires blood for its own tissues, its own tissues. And that blood would be supplied by the coronary artery. Why? Because the blood contains two most important things. One is glucose and the other one is oxygen. And why? Because these two are tickets for survival. If you are not getting it, your cells would die. Tickets for survival. So that is one of the most important things. Now, where these things are basically located, let me show you. You'll be finding the coronary artery on the heart, um, like the yeah. Just look at this. This these are the coronary arteries.
Yeah.
These are the coronary arteries, and we basically are the corona. They basically appear as the um, you can say the crown of the heart. The corona literally means crown. So coronary artery, it's the crown artery. So the appearance of the coronary artery would be like this. Okay. So what happens? Okay, I'll not be drawing the diagram over here. I'll just be taking you to this picture. Just ignore currently. Just don't look at this vessel, the grafted bypass. Now look at the placement of the vessels. The vessels, this is one of the major vessels. One of the major vessels. And these are the vessels which are covering smaller area, covering small area. Remember that when when the blood is not provided, when the blood is not provided, the heart cells, the cells would start dying. And once they would start dying, it means that the heart would start getting weakened. The problem is, this is also the major vessel. This is another major vessel. The problem is the blockage, the position of the blockage matters. Suppose that there is a blockage around here. Let's mark point A. Then there is a blockage around over, let's say here. Let's mark it point B. And then there is a blockage around here. Let's mark it with C. Which blockage is going to be the deadliest one?
The one at C,
Yes. Why would you say that?
because it's basically in the middle, so it would have an impact to basically the whole heart.
Yeah, it would be leading to a major, you can say, blockage, and a lots of heart cells would die, leading to extreme weakness of heart. And the heart cells suddenly shift to the anaerobic respiration. That would also be dangerous. Many people don't directly get the heart attack, but they suddenly start vomiting without any previous reason, without any apparent reason. They suddenly starts get vomiting. Why? Because at the time, you can write that on chat gpt, that why the person with the heart attack sometimes vomit. That is because the heart releases, there are like the dead parts of the cells, the chemicals of the dead cells and the lactic acids are released into the blood, due to which your brain gets, you can say, uneasy and you start vomiting. So that is why a person has to be taking care of everything when it comes to the first heart attack. And the people, remember that this C, the blocking at C, it is simply life taking. It is simply life threatening. Simply life threatening. That is why some people simply die after first heart attack. Like that is they are literally, there's literally heart failure,
Yeah,
or it is also called as ischemic heart problem. Ischemic heart problem. Heart getting less uh, less number of less amount of blood, and the people with having less a damaged area, they can survive. So this is the presence of the arteries. What a beautiful picture, beautiful view is there. All these are coronary arteries, all these are coronary arteries. And you can imagine that this is the main vessel. Look at this. This is the main vessel which is branching. If there is a blockage in the main vessel, then everything is done. Now no chances of survival. Okay, let me try to show you the picture of the heart at the time of angina. Oh, that's not good. Okay, coronary heart, coronary heart. Okay, if we can get some appearance of a clot in a picture over here. Uh, no, human heart. There was a picture, I guess. It was an animation over here. Mm, no. Okay, leave it. But when it comes to the blockage, the heart cells would die, and that part of the heart is typically would appear um, decolorized, or you can simply say um, it would be simply black. It would appear black. So, what are the causes? The clot basically blocks it, and that clot is called as a thrombus. And this problem is called as atherosclerosis. Atherosclerosis. So athero leads to atheroma. Atheroma is a collection of fats with the collection of fats with a lot of white blood cells, with a lot of white blood cells, um, and blood clots. Basically, this is atheroma. The thrombus is only the clot, and atheroma is fats, white blood cells, and the blood clot. So that is basically the overall situation of the atheroma. Why is it collected at the site somewhere in the coronary artery? Remember that the coronary artery, the biggest problem is the high blood pressure. Biggest problem. Because what happens that high blood pressure damages the endothelium of artery. You know the endothelium? Do you remember what is endothelium?
Um, it's the cell lining of the blood vessels, right?
Hmm. Yes, yes, yes. Leading to leading to atheroma formation. Remember that if it is only damaged, there would be thrombus formation. But if there is something called as hypercholesterolemia or high fats in the blood, then there would be deposition of fats beneath the endothelium, and the more the fats, the larger the clot, larger the blockage. And you can simply see the blockage over here. They have shown the blockage. This might be the blocked vessel. So we can do two things. Uh, one is bypass surgery. Like, don't let the blood enter. Don't let the blood enter into the clotted artery. Instead, place another artery, and there are extra arteries in our legs. Place another artery in the heart, and then provide the blood to that grafted artery, bypassing the clot. So that is called as bypass surgery. And the bypass surgery basically happens when the clot is a bit too much large, or it is very difficult to place the stent. But if there is place to if there is some you can say there is a chance to place the stent, then we just puncture the skin, go into the main artery, and with the like the endoscopy, we just go there, and then we just place the stent into the clot, at the place of the clot. So the stent would cover the clot, and it would let the blood flow go smooth.
Okay.
I guess it should be, um, or maybe let's try angioplasty. If there is some sort of information around here. Okay, leave it. So basically, when the stent would be placed, the artery would appear like this. The artery would appear like a balloon. Why is that? Because actually the stent has covered the clot. This is the clot. This is the position of the previous clot. Like it becomes like this. This artery would appear like this, and the place where the stent is, the location where the stent is placed, it would appear like a balloon here, like this. And this is the other side of the clot. And in the middle, now you have enough blood and enough space for the blood to pass. Now, what are the reasons of this problem? Now, this is something to understand very clearly. Okay, first is poor diet, high levels of cholesterol or saturated fatty acids in the blood. Remember, uh, it may be taken from the refined oils. The basic dangerous thing is the refined oils. Always remember, the animal fats is not the problem of the cholesterol. Is not the cause of the cholesterol. It's only the refined fats. Okay, the second is poor lifestyle. Someone is smoking a lot. Smoking causes the clotting.
Yeah.
So that is basically very important to know. Then lack of exercise and stress. Stress also thickens your blood, so that is also there. Then genetic factors. Genetic factors is the problem here. Basically, the gender is the most important thing. Males, due to their primary hormone, testosterone, tend to accumulate more fats. Due to testosterone, tend to accumulate more fats and more clots, tend to accumulate more fats. Then having a family history of heart disease, that is another problem, and a very big problem here. Okay, the person who basically has the family history of heart diseases has to take care of himself or herself very much, very much, because only one heart attack can just take his life. Got that?
Yeah.
Okay, so that is not the chapter we'll be talking about. Okay, all right. Okay, so now I'll just hang on. Gas exchange, respiration, excretion. I'll just be jumping to another chapter, the excretion. That chapter is smaller, and we can just finish it. Okay, what do you know about this chapter? What is excretion?
Um, the release of waste.
What wastes?
So waste from food.
Mhm. First, make it very clear that excretion does not involve the food wastes. It only involves the getting rid of carbon dioxide and getting rid of urea. Urea is basically made during the protein metabolism. So that is excretion. CO2 obviously would cause the suffocation. The brain would not perform properly in the presence of too much carbon dioxide. When it comes to urea, urea is basically least poisonous form, least poisonous nitrogenous waste. Still, if there is high amount of urea in your blood, it can cause oxidation in your body, oxidation of molecules. It can cause the coma, it can cause the heart attack, it can cause the death if the urea accumulates in your body. Simply putting it, given the fact that it is the least poisonous nitrogenous waste, the formula for urea is this: NH2, NH2. This is the formula. The most dangerous form of nitrogenous waste is ammonia. And what happens, basically, what happens, that in the liver there is a cycle happening called as uh, the urea cycle. The urea cycle. What happens that we have an amino acid. If the amino acids are in very high amount in our body, the problem is we cannot store we cannot store nitrogenous compounds. We cannot store nitrogenous compounds. We can store the fats in our body.
Yeah.
Can we store the carbohydrates in our body?
Yes.
But we cannot store the nitrogenous compounds. We cannot store the amino acids in the form of amino acids. We have to get rid of the amine group. This group has to go. What happens that we add one amine group here and another amine group here, and we get the urea. This process is occurring in the liver. We just get rid of the amine group. The rest of the we do not have a problem with rest of the group. We do not have the problem with rest of the amino acid because the amine group is released. Now this liver would transfer this urea in blood towards heart, and the heart would pump the blood, and from the heart it would go to kidney. Not only kidney, obviously it could also go to the brain, but its amount are not poisonous for the brain. The amounts that are being made when our liver is working properly and the kidneys are working properly, then the amount of urea in our blood does not matter that much. Understood?
Yeah.
Now, how do we basically remove the urea? We remove the urea with the help of urine. So we have two systems running in our body. One is called as excretory system. Excretory system. Excretory system involves the liver, the kidney, the ureter, the urinary bladder, and urethra. And other than liver, with liver it is called as excretory system. Other than liver, it is called as urinary system. This is the difference between these two systems. Got it?
Yeah.
Let's open the new Jamboard. You should copy the link. I'll be opening its sharing. Any problem till here?
No.
You can access it now. Okay, now let's see what happens basically that when it comes to the urinary system. When it comes to the urinary system, you also know that urinary system has has one major organ that is present in the pairs, and that is called as the kidneys. The kidneys are bean shaped. The kidneys are bean shaped, and you can see three types of vessels entering and exiting from the kidney. One is this, the other one is this, and the third one you'll find is on the back, this one. Now, the first one, the red one, is the renal artery. What is the meaning of it? Containing a lot of urea. What is the meaning of renal artery?
Um, supplies blood to the kidneys.
Hmm. Then the blood. Then the second is the renal vein. It contains least urea. Why do you think this happens? That renal artery contains a lot of urea, while the renal vein contains least urea or no urea. See, when the blood enters from the renal artery, it goes all around the kidney, gets filtered, gets filtered. Now, all urea would go to ureter. This is ureter, and it is containing maximum amount of urea. All urea would go to ureter, and the blood would enter into the renal vein. That is why the artery was containing the urea, but since it got filtered, now the urea is in the ureter. The rest of the cleaned blood would be flushed into the renal vein. Got it?
Yeah.
So that is how the this phenomena happens. The problem is like how the kidney does it. How the kidney does it. How the kidney makes it possible. Kidney does it. What is working inside the kidney? What is the most important part? Now, what do? We section the kidney. And what section do we take? We take longitudinal section. What is longitudinal section?
So the vertical section.
No. Vertical section is transverse section. The longitudinal is along the length.
Okay.
So we cut the kidney along the length, and we get similar appearance of the tissue, whole tissue, and we observe it under the microscope. It seems that the kidney is divided into two portions, and basically that is because of the color. The outer red portion, the outer red portion is called as the cortex. The red portion is called as the cortex, and the red color fades when we move inside the kidney. It fades and it becomes a bit yellow. It becomes a bit yellowish. This is called as the medulla. The outer part is the cortex and inner part is the medulla. Now, if we observe it under the microscope, we see some circle-like thingies over here, some circular structures in the cortex. Now, what are these? Now, you have to magnify it a bit more in the under the microscope. Now, when you magnify it, you come to know that okay, uh, this is the cortex and this is the medulla, and this circular thingy that you are looking at is basically the initial part of the main tissue of the kidney, and that main tissue is called as what? That main tissue, what do we call this main tissue?
I don't know.
Nephron. Nephra or nephron literally means kidney. It literally means kidney. So basically, this is the nephron, and you can simply say the nephron is the functional unit of kidney. What is the meaning of functional unit, by the way?
The standard unit.
You can say the standard unit with respect to function, or you can say the unit that represents the function of the kidney. Like you have a shoe factory, suppose, and you have 1,000 machines to cut the leather or maybe to make the shoe. Now, do you study all of them? No. You just take one out of it, assuming that it is representative of all those machines, and you study it. Similarly, the nephron is the functional unit of kidney. Basically, it would represent all the tissues of the kidney. Remember, it's a tissue. Okay, so now we'll magnify it more, trying to understand what the nephron is. Okay, I'll tell you in my own way. Now we'll be magnifying the nephron. When you see the nephron, you'll find two main parts. One is called as renal corpuscle. Renal corpuscle is basically the literal meaning of, let me show you, what is the literal meaning of corpuscle. Body or small body or cell. Basically, it is, you can simply say that renal corpuscle means it's a body that you are finding inside the kidney. So renal corpuscle is a part of the kidney, which is this one. This is renal corpuscle. If I mention this one, so this is one, only this region. This is renal corpuscle. It has two subparts. One A is called as the Bowman's capsule. Bowman's capsule. This one is 1A. And 1B is the glomerulus. The glomerulus is basically the cluster of capillaries. Cluster of capillaries. So basically, where would you see it? You'll simply find it out over here. These are the capillaries you'll be finding out. So basically this is 1B. So renal corpuscle is this whole body. Bowman's capsule is the blue part, and the glomerulus, basically from here the blood reaches to the nephron, and from here or you can say renal corpuscle, and from here the blood leaves. That is why this one is named as efferent arteriole. Efferent means arriving, arteriole is smaller artery, and this one is called as efferent arteriole. Efferent means exiting, like blood enters from efferent and leaves from efferent arteriole. Both are arterioles, remember. None of them is vein or artery. Both are arterioles. This is arteriole and this is arteriole. Okay. Any problem till here?
No.
Then comes the part renal tubule. Tubule basically refers to small tube. Tube is for the large diametered vessel. So there are two types of tubules you'll find in the nephron. One kind of tubules are convoluted. Convoluted mean folded. So if I say the renal tubule, let me mention the renal tubule as two. So convoluted tubules would be named as 2A. And the other type of tubules, they are not folded, but they are looped. So we'll be calling them loop of Henle. Remember, both these are tubules, but because of their shapes, we name them differently. Convoluted are folded ones, the other ones are looped ones. So I would call the looped one as 2B. Now, obviously, the convoluted tubules are further divided. One are called as proximal. Okay, what is the meaning of proximal?
Like in close proximity.
Close proximity. Very close to a reference point, if I'm not wrong. So proximal, this is the, I would mention proximal as 2AX. So this one is the proximal. Proximal convoluted tubule. The complete name is proximal convoluted tubule, or PCT. And then there is another convoluted tubule called as distal. Distal means far from a reference point. I'll mention it with 2AY. Now this is basically 2AY. Distal convoluted tubule. It is also folded. I made less foldings here, but it is also folded. So 2AY, this is distal. Distal to the reference point, that is renal corpuscle. Renal corpuscle is the reference point of a tube that is folded, and closer to the renal corpuscle is proximal convoluted tubule. And the tubule that is folded but at a distance of the renal corpuscle would be called as distal convoluted tubule. Understood?
Yep.
Now, loop of Henle. Loop of this one, this whole is basically 2B. Loop of Henle. We divide the loop of Henle into different, you can say, into different parts, but that is not basically the point of the discussion. So I'll not tell you the further parts. Now, the real part is how the nephron works. How the nephron works. Basically, the first part of the nephron is filtration. The first thing of the nephron is filtration. Filtration means you are going to filter out the substances. And this thing happens uh, with the help of glomerular capillaries. Why? Because capillaries have pores. The pores of capillaries act like the sieve of the filter. Capillaries act like sieve of the filter. Like, you understand? What is the
Yeah.
sieve of a filter? So basically, it has small pores. So it acts like the sieve of a filter, and the things would be filtered out from the glomerulus. So, but uh, the important thing to understand over here is that the all small compounds would be filtered, would be filtered, like water, glucose, amino acids, amino acids, and then the what else? Yeah, urea, electrolytes. But the large compounds would not be passing through the capillary pores, be passing through the capillary pores, capillary pores. What large compounds are not going to pass? Uh, Blood cells and proteins. And always remember, this filtration is the pressure filtration. What is the meaning of it? The pressure inside the pressure of blood in glomerulus is higher than any other capillary, which typically means that which typically means that the pressure would cause the filtration. It's just like you have a sieve, and and the water or the blood is coming with a particular pressure colliding with the sieve. What do you think? That collision would cause the filtration or not?
Maybe.
Yeah, it would cause a lot of collision because when the blood is coming with the pressure and a speed, it would collide with the capillaries, leading to the leakage of all the substances that are smaller enough to pass through the pores. Did you get it?
Yeah.
Okay. Now, this is the first thing, and it basically happens in the glomerulus. Like, this is the Bowman's capsule, and and the capillaries that are coming here, that are present in here, these capillaries, there are arteries. Sorry, those are arterioles, and then these are the capillaries. The blood goes there, gets filtered. This is now the lumen. This is called as tubule lumen. So everything goes into the tubule lumen after getting filtered. So the things are filtered now. But what about the substances that we need? Many substances have been filtered. Sorry. Many substances have been filtered that we need. that we need. Now, how do we do that? Like, we have to get them back. To get them back, we do the phenomena of reabsorption. Why do we call it reabsorption? Reabsorption, and not the absorption. Hmm?
Um, can you repeat the question?
Why do we call it reabsorption and not the absorption?
Because it's been absorbed before.
Where?
Um, I don't know.
We have absorbed all these things from the intestines. Don't
Okay.
you think?
Yeah.
In the intestines, we absorbed our food. So that basically is the uh, absorption. And now, once we filtered the things, now we are getting them back into the blood again. That is why it is called as reabsorption. Why did I write 2 here? This is the second step to be occurring inside the nephron. The reabsorption occurs in the proximal convoluted tubule. Basically, what I mentioned over here is 2AX. It occurs in the proximal convoluted tubule. What things are going to be reabsorbed? The glucose. The glucose is 100% reabsorbed. Amino acids, they are also 100% reabsorbed. Water, 70% reabsorbed. Then electrolytes, they are also 70% reabsorbed. We get all of these things back immediately. But urea is not absorbed. Urea is not reabsorbed. Why? Our main purpose is to get rid of it. It is not reabsorbed. Now, uh, after this, what comes? That is the loop of Henle. Loop of Henle, or you can say 2B that we mentioned over there. In there, we reabsorb a bit more water and sodium only. What are you clicking on? Hmm? What are
Hello.
you? What are you? Yeah, I was saying. I was asking. What are you clicking on?
I was trying to unmute. I was about to ask a question, but you answered it. Never mind.
Okay, I have already answered it.
Yeah.
Okay, so in proximal convoluted tubule, everything would be majorly reabsorbed. In the rest of the in the loop of Henle, we reabsorb a bit more water and sodium only. What is the main purpose? We are going to concentrate urea. We are going to concentrate urea so that urea could be released out. Could be released out. Now, at the end, now I have missed one thing over here that I will tell you now. One part of the nephron that is not related to only one nephron is this. Hold on. The last part of the nephron that is not related to only one nephron is the one I'm drawing. This part is called as collecting duct, and it basically collects the urine from 8 different nephrons. 8 different nephrons. So basically, after the reabsorption, after the reabsorption, after reabsorption is done, the urine, the urine now in bracket, please focus. The point that I'm about to write in bracket is the primary point of this line, chiefly consisting of a large amount of urea, extra water, extra water, extra sodium, and some other wastes. After reabsorption is done, the urine is flushed into ureter, stored in urinary bladder, urinary bladder, and released through urethra upon proper stimulus. Upon proper stimulus. So this line is very important, chiefly consisting of large amounts of urea, extra water, extra sodium, and some other wastes. Did you write it all?
Yes, wrote it all.
Good. Okay. The role of glomerulus, the role of nephron in the reabsorption, formation of urine containing urea, excess water, and excess ions, the role of liver in assimilation of amino acids and converting them to proteins that we have already discussed. Urea is formed by the liver, describing deamination, importance of excretion. What is deamination? The last point. The deamination basically removing the amine group from the amino acid. This process, this is called as deamination. And always remember, it only occurs to extra amino acids. If the amino acids are just sufficient enough to make the proteins, they would not be converted into urea. If they are sufficient enough, if they are extra, they would be converted into the urea. Is it clear?
Yes, it's clear.
Anything that bothers you?
No. So that was past the heart topic, right? Now we're on the kidney and topic, or is it all in one
Yeah,
topic?
heart topic was finished, and the excretion topic is also finished.